Good morning, good afternoon, good evening, wherever this finds you. Welcome to the 87th Emerging Growth Conference and day two of our Super Virtual Investor Conference. I'm Anna Berry. Just a few notes. Today we're running until about 4:50 Eastern. And when we switch to the next company, you'll see a black screen for a moment. Don't go anywhere. That's just us moving over to the next presenter. But if you do experience downtime, refresh your browser. Everything usually works properly once you do that. And our platform does work best on Google Chrome. So if you're watching from an Apple device, you have to hit the play button to start the session. Now, during each company's presentation, you can submit questions through the webcast module, and we will attempt to address as many of these at the end of the presentation. All of our conferences, they're uploaded to our Emerging Growth Conference YouTube channel, so please subscribe. And one last note, after today's event, you'll be redirected to the registration page for our next conference. So stay on or come back to reserve your spot early. Let's begin. We have Imagion Biosystems Limited, trades on the CXA under the symbol IBX and on the ASX under the symbol IBX. They are developing a new non-radioactive and precision diagnostic molecular imaging technology. Combining biotech and nanotech, the company aims to detect cancer and other diseases earlier and with higher specificity that is currently possible. Happy to welcome Executive Chairman Bob Proulx and Chief Business Officer Ward Detwiler. Welcome, gentlemen. Happy to have you on the conference, and we're very much looking forward to hearing your presentation.
So we're a clinical stage company developing a new class of molecular imaging agents for the detection of cancer. We've got a pipeline that we'll talk about today, and we got a couple of important upcoming key milestones. But starting with the 50,000-foot view of things, I think it's important to try to characterize really what it is that we're going after here. I think everybody's familiar with and unfortunately touched by cancer in some way, shape, or form. 1 in 3 people typically either are directly affected or have somebody close to them that is. And what's probably not known frequently is that we spend about $100 billion a year globally to try to diagnose cancer. And yet we don't seem to still be doing a very good job despite improvements that we've had in detection technologies. The graph on the right sort of shows the impact of not being able to, wow, why did that jump ahead? The graph on the right demonstrates that if we catch cancer early, our ability to treat it is very good, but if we catch cancer late, then the outcomes are not very good. So our focus is really on what can we do to do a better job when trying to diagnose cancer, to catch it early and be specific about what it is that's really going on there. And so, the really unmet need then is this $100 billion a year that we spend to try to diagnose cancer is really focused in a couple of different areas. We've got screening tools such as blood-based tests that indicate you might be at risk for something. So for men, think about a PSA test that indicates whether or not you might be at risk for prostate cancer, or a mammogram for women to look at whether they might be at risk for breast cancer. But invariably, we still have to do a biopsy. The way that we actually diagnose the presence of cancer is by actually doing an invasive procedure, sampling the tissue, and being able to then look and determine whether or not it's actually cancer. And that's because current imaging methods don't actually have the ability to be specific. They are able to look and find something that might be of interest or something known as being suspicious, but they can't tell from normal imaging whether or not it's benign form of tumor or whether it's a malignant form of cancer. And that's the problem that we're trying to address. With our technology, we have an imaging agent that's highly specific for various types of cancer. And that is detectable by MRI. And so when you see our particle present, it's there, and that suspicion is moved into certainty because the only reason the particle is there is because it's directed to a cancer cell. We don't use radioactivity as the tracing mechanism. We use a small magnetic particle, which works with the existing installed base of MRI systems, and it's applicable to a wide variety of different types of cancer. So how does that actually work? So we take, as I said, a tiny magnetic particle, and we attach various types of targeting molecules to it. So in the case of breast cancer, we'll use an antibody specific for that form of breast cancer. In the case of prostate cancer, we'll use a peptide that's highly specific for prostate cancer. etc. So our particles are highly specific for the particular type of cancer that we want to be able to detect. And that's what gives us the specificity and the certainty of when we see our particle present, we know it's there because of that targeting molecule. So those particles then cause our particles to circulate and then bind to the tumor cells if they're present. And they produce, when present in the tissue, they produce a very identifiable kind of change in contrast. And what you see here on this image on the right is, in fact, the change that happens. So the image on the left here is the pre-dose image of a woman's lymph node. And the post-dose image on the right shows about a 30% change in contrast associated with the presence of our particle. And it's this change in contrast that's exhibited by the presence of our particle that is reviewed by the radiologist and completely identifiable by them. So instead of a radiologist looking at this and saying, this node looks suspicious, it's enlarged, it's a little irregular in shape, they now have the certainty to say, not only do I have anatomical information that tells me that something that might be amiss there, but I can actually see the presence of the particle, and those particles are there because of the specificity of the targeting molecule that's on them. What's nice about the use of the particle then is it can also be used with artificial intelligence and machine learning algorithms to improve the radiological review. So here you can see, for example, the actual original MRI image of the lymph node. Then they're able to hone on that image of the lymph node itself and look at that heterogeneous or spotty pattern associated with the presence of the particle. So pixel by pixel view of the lymph node. And then here is for example, is an algorithmic solution that looks at that lymph node and compares the homogeny of the pre-dose image to the spotty pattern associated with the post-dose image. And so algorithms can be applied to be able to interpret this and aid the radiologist in their review of this. So we know that moving forward, we'll have the ability to combine the use of our particle with artificial intelligence or machine learning algorithms to really assist the radiologist in the review and move from being able to look at an MRI image and say something looks suspicious to be able to then actually have certainty that they're with what they're looking at. Is that really a malignant form of cancer? What's nice is, as I said, we have a pipeline of products and they all work the same. So here's preclinical model of an ovarian cancer, and you see that same change in hypointensity or that same change in contrast from pre-dose to post-dose. And so this particle was designed specifically to target ovarian cancer cells. And so what we have then effectively is not a one-trick pony of a breast cancer application, but we've actually got a platform with our particles that allow us to develop individual cancer-specific imaging agents that can then all work in the same way. So let's take a look briefly at what that pipeline of products really looks like. So we'll go through each of these use cases a little bit briefly here. Our first and lead product is for the nodal detection of nodal metastasis in HER2 breast cancer. So the problem that we have today with breast cancer is not so much the identification of the presence of it. Mammograms are a pretty good screening tool, and we can then do a biopsy to determine whether or not that tumor seen on a mammogram is actually cancer. But the problem then becomes, if it is in fact cancer, the first thing you want to know is, has it spread to the lymph nodes? Is it metastasized and begun to spread, or is it isolated to just that primary tumor? Because that affects how they think about they're going to treat the patient. And so today we have the standard of care being to use ultrasound and biopsies to try to determine whether or not a woman's lymph nodes have had metastasis to it. The problem is that about 50% of the time, the answer is no. So the doctor gets to go back to the patient and say, the good news is no metastatic spread. The bad news is I just had to do a biopsy on your lymph nodes to try to determine that. We look at that as an opportunity to try to eliminate and minimize the need for biopsies, because if we could non-invasively determine whether there was nodal involvement, it'd be much better for patients. Anyone who's familiar with someone who's had a breast cancer and had a lymphedema, which is an after side effect associated with doing biopsies on lymph nodes, we know that we have the opportunity to try to reduce the false negative and false positives by non-invasively using imaging. That same kind of approach works for prostate cancer. If you think about men having a PSA test, but then if it's an elevated PSA, in most countries around the world, they'll revert to doing a prostate biopsy. Well, unfortunately, we have about a 50% false positive rate. That is 50% of the time, MRIs indicate that there's something that's there. When they go do a biopsy, it turns out that it was false. It wasn't actually a prostate cancer. PSMA PET is increasingly being used, but it's typically only being used for the detection of metastatic tumors. So again, here we look at the opportunity that if we could take a person who has an elevated PSA blood count and not do a biopsy, but first do a MagSense test to determine whether or not we see that contrast associated with the presence of our particles, we could then use the presence of our particles to do an MRI-guided biopsy and reduce the false positive and false negatives that you see from just standard biopsy procedures. The one that's near and dear to my heart actually is the ovarian cancer case, and that's because we don't do a very good job of diagnosing ovarian cancer. 75% of the cases in ovarian cancer are diagnosed late stage, and that's because today the standard of care is to do a blood test for women who are at risk for ovarian cancer. But if you see a CA-125 blood test, you typically do what's known as watchful waiting by ultrasound. That is, about every 6 months, the patient gets brought back in, they do an ultrasound to see if in fact they see a tumor growing in and around the ovaries and the uterus. The problem is that the standard of care actually requires that ultrasound, because ultrasound's images are not high resolution, it actually requires about a 5-centimeter mass before they're willing to determine that in fact that tumor needs to be addressed. And so by the time an ovarian cancer is about 5 centimeters, it's usually a stage 3 or stage 4. And as we saw from the earlier graph, outcomes for patients at late stage cancer diagnosis are actually quite poor. So if we could, in fact, take a woman who has an elevated CA-125 blood test and now do a MagSense test, and with high resolution MRI, be able to see if our particles are present because they have a highly specific targeting ligand for ovarian cancer, we would have the opportunity to perhaps be able to identify a patient who has ovarian cancer way before it becomes 5 centimeters in size and be able to then be more treatable. So we see each of these as examples where specific detection will improve the way we're able to diagnose the presence of cancer, eliminate the need for biopsies, and eliminate the false negative and false positives that you often see causing patients the discomfort and stress associated with just getting to a diagnosis. So this is what our clinical development plan looks like and the key milestones that will be forthcoming as we go through this. So we're currently, as I said, our lead product is a HER2 breast cancer product for nodal metastasis. Right now, we are in the process of being able to file our IND. So our phase 1 study was completed in 2023. We did about 13 patients in that study, demonstrated that it was safe and detectable. So we're now moving into the stage where we're moving into a phase 2 study. We'll be submitting our IND to the US FDA coming up here before the end of the year, and we plan to start that study next year. We'd like to be able to begin funding the ovarian cancer and prostate cancer projects as well. Those, all the preclinical work on those has been done. The next thing we need to do then are what are known as the IND enabling studies. Those are the toxicology studies that support your ability to then go into human testing. And so over the next period of time, our development plan is to proceed with the phase 2 study for our lead product and be in a position to have the other 2 imaging agents for prostate cancer and ovarian cancer in the clinic. And we think it's important to be able to get through this because from an overall company perspective, having 3 products in the clinic versus just sort of the one-trick pony approach, we think is what's really going to drive shareholder value. From a business case perspective, this makes a lot of sense in the realm of what's going on in MRI. So we know that imaging is being used more and more frequently, but the problem is that some of the imaging technologies can't do what our technology does. And so we're really focused on how do we then bring that to the market and what's it going to look like from a implementation perspective. So we've already been looking into the health economics of how our our technology would work. You can see the graph on the right that comparing standard of care to the use of our MagSense technology, we save about $1,500 per patient every time that they would do a MagSense test versus doing the standard of care. What that does is it drives the ability for us to potentially then go get a value-based reimbursement strategy and a dedicated code compared to what you would get if you were to just use standard contrast-enhanced MRI. Our products have a long shelf life, so they fit within the existing workflow within the hospital system. Unlike a PET imaging agent that needs to be made on-site and used immediately, our products are able to be stored and sourced at the hospital pharmacy. So if a patient came in, the doc would be able to go ahead and request the MagSense test directly because the hospital would be able to have the test on hand. And as I said earlier, they fit within the existing workflow of MRI and will work with the existing 50,000 plus scanners that are in use globally. So we see this as being a transformative technology because not only will the algorithmic solutions make diagnostic accuracy better, but it works within the existing hospital infrastructure. Oh, and that's really important from an overall business perspective for us because our business plan has always been to partner with someone in the marketplace. There are a number of companies that already are in that space, and rather than think that we're going to go to market and develop our own commercial organization, we see that space as being best for partnering. And what a partner is going to be interested in is that fits the business model that they have. You have multiple products available in the clinic. And so we see each of those three things that I was describing earlier as being the substantiation for why we think a partnering approach is going to work for us. And the question really ultimately then is, when is the appropriate time for value for our shareholders to be able to go get value from a strategic partner? But we know that the partners are interested, the companies in that space are interested in expanding, and we have something that doesn't exist. This is, we're doing something that's first of its kind in medical imaging. But from an overall investment rationale perspective, we see this as being, we're addressing large markets, and to date, the current methods don't address what we address in that by using our technology. It's a relatively, from my perspective, at this point in time in the development of the company, it's becoming a lower and lower technical risk. About almost 100% of contrast media products are actually approved by the regulators. And that's because unlike drugs that are trying to treat cancer, we don't have to cure the cancer to get to an efficacious outcome. We've only got to be able to demonstrate that, in fact, we detect the cancer that we say we're going to detect. And so contrast media products are typically approved by the regulators. So we don't see the regulatory environment as being a significant hurdle. It's something we have to go through, and it costs money to do that, but it's the phase 1 data that we have already support the fact that the product is safe and detectable. So again, we've taken a lot of the risk out. And as you saw from an earlier slide, all of the particles work the same. So that bodes well for our ability to get each of the products individually through the clinical process, because in fact, we have precedent each time we go through, we have the precedent of the prior product going through the regulatory process. And so, as I've said, it's a commercially attractive for strategic partners because the health economics support the value-based reimbursement strategy. The product itself will have high gross margins, which will support us working with a strategic partner. And so we think that we've got a business plan here to bring value for our shareholders because we have something that's unique in the marketplace that addresses large market opportunities, has a likelihood of being successful, getting through to commercialization. And our job now is to continue to fund the development of that product such that we can go and then commercially partner with somebody. Just briefly on sort of the overall for the company, we've got good leadership of the company. Ward's on the call with me. He's a chief business officer. His background is in MRI. He was CEO of a company that was developing algorithmic and computational methods for MRI. So he's got a good background there. Our board, Nina Webster in particular, is an experienced CEO, background in pharmacology, developing drugs. So we've got a good board, an advisory board with Susan Harvey and Leo Kayat-Bittencourt advising us on the clinical side of our products. All of us in here have experience in commercialization of products and going through the regulatory process. So I think we've got a good board that's focused on this strategy for the company and supportive of the overall strategic plan that we've got in place. And just as a final slide before we go into a Q&A session, just the highlights for the company. We're addressing about $2 billion worth of market opportunity. Our product is first of its kind, so it's quite unique. And we believe that the strategic partnering commercial model is actually best for our shareholders, because with the strategic partnering model, we should be able to attract non-dilutive funding to help defray some of the costs of getting there. We are currently operating as a virtual biotech, so our G&A costs are quite low. And we plan to continue to do that, keeping our staffing and facilities costs at near minimum while we're going through this clinical phase. Since most of the clinical work is outsourced as you're doing these studies, we're going to try to keep that virtual biotech organizational structure in place and keep our overall costs low. We're traded on the ASX as IBX. Most of our shareholders are retail shareholders. We've had good liquidity this year, with about 3% of shares traded on a daily basis. So new investors have the opportunity to come in and know that, in fact, there's some liquidity to the stock. We've got about $3 million in cash on hand as of September 30th. Those funds will be used to help us get started, file the IND, get started on that phase 2 study, and initiate some of the IND-enabling studies for prostate and ovarian cancer. And with that, I will go ahead and open the Q&A session.
Perfect. Thank you, Bob. Thank you, Ward. All right, let's start with how has Imagion looked into what reimbursement might look like?
Yeah, so I think on that slide, we've actually did the health economic study, and then we did the separate reimbursement. So in the United States, which is the largest market that we would be addressing, the whole CPT coding is the way it's done. And in single payer markets like the National Health System in the UK, and it's a little bit different in Australia, there is a reimbursement coding in Australia. But what we've looked at there is that because it does the specificity, it gives you a specific answer as opposed to something more general like gadolinium-based contrast agents. There is the route in reimbursement coding to be able to get a specific code for our product. And so what we've anticipated is that we would be able to go get a dedicated CPT code for the MagSense technology in general, and that would be able to get reimbursed at around $1,200 to $1,500, because in each of the use cases that I described, the health economics work out that we're actually saving more than $1,500 per patient by doing the MagSense test compared to standard of care. And so by getting a dedicated code that's still less than what the healthcare savings are, the overall system benefits and then the margins on our product are attractive, because at around $1,500, we'll have about a 90% gross margin on our product.
And how will this tech play with AI and machine learning?
And so I'm going to let Ward field that, since that's his primary area of expertise. And I'll go back up to that slide for you, Ward.
Yeah, so this is a really exciting area for us that I think draws on some of the past work that I've done. First of all, when we look at the images themselves, radiologists, academic medical centers, the people that have been working with the studies, they clearly see this difference in the pattern between an affected and a normal node. But radiologists are extremely busy. They are incredibly overworked, and not all of them have specialized training. So we see AI as an important way for sort of automating the detection process of this, but there's some challenges within that. MRI is still a kind of a subjective tool, largely, where you're looking at changes in contrast and kind of making a judgment decision. So it's sort of like a bit of a garbage in situation for developing these AI algorithms. By introducing the specificity of the molecule that we have, along with introducing quantitative imaging techniques that we're developing with Wayne State University and some leading MR physicists there, we're able to provide some really high quality data to go into these models so that they're actually providing very accurate results that not only allow us to provide automatic, identification of effective nodes, but also provide a kind of a host of other data in there around staging and looking at progression of the disease itself, looking at treatment effectiveness, So there's some really exciting things that are going on right there. We're expecting some results of those studies pretty shortly. But this is, for me, a very exciting development for us, and something that really think can kind of unlock the key for AI and MRI.
And can you talk to us about any patents? Have any patents been issued to you to date?
We did have a patent portfolio based on a different method of detection. We're in the process of some patent applications that will tie the particle to this sort of AI approach of detection. So we see there's an opportunity for, it's not, the particle itself is not patentable because iron oxide is a standard type of imaging tool. But what we see the opportunity then is this combination of the imaging agent with the AI to be able to get some intellectual property. So we've been pursuing that since we started to get the clinical results from the phase 1 study. But the patents have not been issued yet.
And is it reasonable to believe that your imaging technology will eliminate the need for invasive procedures to confirm if a mass is benign or cancerous?
In the long run, that's what we're aiming for. I think to begin with, we will go after, let's say, a lower hurdle or a lower bar from a clinical perspective to get it approved by the regulators. But ultimately, we'd like to see that our data support the idea that, for example, that if a radiologist sees that, they don't need to go do a biopsy because, in fact, they know that there's tumor involved in that node. And that will simply come by more and more data that support the fact that the radiological review is what's known as being concordant with the pathology. To be able to get all the way to an initial diagnostic indication would require thousands of patients. And so what we're planning to do is to start to get regulatory approval for it being used as an aid in the diagnosis. And then as the data eventually come in and support it, we would eventually modify the claim to be diagnosis and replace the need for biopsies.
Perfect. And at what stage would your technology pick up the status of any mass?
So we have demonstrated in the phase 1 study that the particle present in a lymph node is detectable in as small a lymph node as 1 to 2 millimeters. So standard of care today for the diagnosis of normal tumors is about a 1 centimeter mass. So we've actually demonstrated that we can detect the presence of our particles in lymph nodes as small as 1 to 2 millimeters, so 1/10 the size of the normal standard of care. So that's what gives us hope that we'll be able to actually do earlier detection, because if we could get to today, if you saw a 1 millimeter mass by contrast enhanced MRI, it would not be clinically actionable. They would say, well, we don't really know what that is. That's just 1 millimeter of a spot. Whereas if we could say particles are present, then we have an opportunity to actually take action potentially earlier. So we're hopeful that, in fact, our level of sensitivity and the high resolution of MRI will combine to be able to start to detect smaller and smaller tumors. And then, as the previous question asked, potentially be able to do that actionably without having to do an invasive biopsying procedure.
Perfect. Well, gentlemen, do you have any closing remarks for our viewers today?
No, we're very excited and appreciate the opportunity to present to your audience, and we'd love to have anyone who wants to follow up send us directly and invite you to become shareholders of our company.
Wonderful. Thank you, Ward. Thank you, Bob. We appreciate this and hope to see you again real soon with some updates.
Great. Thank you.
All right, everyone. We'll be right back with our next presenter.